Alright, welcome back fellow scientists. So last video we looked at the basic process of DNA replication. What I want to do with this video is go a little bit more in depth and talk specifically about why we say that DNA polymerase adds nucleotides to the 3' end of the growing strand. So if you'll remember, each strand of DNA has two different ends. There's the 5' end with the phosphate group, and then the other end of the strand is the 3' end that doesn't have the phosphate group. Notice also that DNA is anti-parallel. This 3' end over here is directly across from this 5' end right here. Right? If you look at the pentagons and you look at the oxygen, all of these oxygens are pointing up, all of these oxygens are pointing down. So as we add nucleotides to the DNA strand, we're basically building a strand of DNA. Now, if you'll remember from Chapter 2 and Chapter 8 and Chapter 9, when we're talking about entropy and building and chemical reactions, we know that it takes energy to build something, and energy is released when something is broken down. Well, the most famous energy molecule is ATP, and we happen to have that right here, adenosine triphosphate. You say, Mr. Leo, that looks suspiciously similar to all of these molecules right here. Yes, it does, exactly. Adenosine triphosphate, ATP, is a nucleotide, right? It's a part of DNA. It's also the energy currency of the cell. We could have GTP, guanine triphosphate, CTP, cytosine triphosphate, TTP, thymine triphosphate. ATP is a nucleotide. It's just the most famous nucleotide, so it's a very energetic molecule, right? So if we add ATP to the 3' end of the growing strand, because it has 3 phosphates, it brings its own energy. So what DNA polymerase can do is it can take this nucleotide, it can break off these 2 phosphates, and then it can add this phosphate to this sugar right here, and lo and behold, now this nucleotide is a part of this growing strand. And notice the pattern is continued. Sugar, phosphate, sugar, phosphate, sugar, right? Now, what happens if we were to try to add this to the 5' end of the strand? Well, notice that there's only one phosphate group right here, right? And so we would have to flip this, and this would have to be in this orientation because the base has to be facing towards the middle, right? DNA polymerase, there's no energy right here to add this nucleotide, to build this nucleotide onto this strand right here. DNA polymerase can't use this energy. DNA polymerase has to use the energy of the phosphate that's going to be a part of the chain, right? So the reason why nucleotides aren't added to the 5' end of the strand is because that would rely on the strand for the energy, which ATP is highly reactive, so these phosphates get used up, or they go away very quickly, very readily. And so this nucleotide would have to rely on the strand to provide the energy, which isn't always going to be there, and if that happens, then the strand is just stopped being built, right? But if we add it to the 3' end, the nucleotide is bringing in its own energy. And so let's say, for instance, that this nucleotide it's not a triphosphate, it's a monophosphate, and it somehow gets in here, and DNA polymerase says, oh, I can't use you, so it just kicks it out, and then it waits for one that does have 3 phosphates to come in. Like this, and it says, oh, okay, sweet, I can use this one. So the strand doesn't stop being built, it just waits for the right thing to come in, and the strand continues, the pattern continues. So that's why 3' is important. Now, let's look at what it looks like using other drawings. So again, here's ATP. Remember, this is the 5' end of a nucleotide. This is the 3' end of a nucleotide. So we're going to shrink this down to size so we can actually fit it into our drawing, and then we're going to rotate it like that. Remember, 5' up here, 3' right here. So DNA polymerase is going to start here, and it's going to take this, and then highly reactive, so that goes away. And then we're going to have another one that's going to come in, remember, adding to the 3' end. So then we're going to use that energy from ATP. Those are going to go away. That energy is going to use that to build that, and then we're going to have another one come in, so on and so forth, and then that's going to go away, and we're going to use that energy to build that, and then we see this pattern: sugar, phosphate, sugar, phosphate, sugar, phosphate. All right, sweet. So now we can label 3' and 5', right? So we're adding to the 3' end. So that's 3'. That makes this has to be 5'. Okay? So now if we follow this, as DNA helicase unwinds the double helix and as DNA polymerase makes this strand grow, eventually this end of the strand will be opposite this part of the strand right here, because these two strands are anti-parallel. So if this is 3', eventually this is going to grow, so that makes it so that this has to be 5'. Well, looking at just our original DNA, if this is 5', that means that this has to be 3', right? So then we can take this and we can follow it all the way back. If that's the 3' end of that strand, then this has to be the 5' end of this strand, and because our original strand, just like our new strands, are anti-parallel, then this has to be the 3' end. And then here's where we really see if we did this right. Then we look at our other new strand. So the 5' end is over here. Eventually this is going to grow and it's going to be opposite this, so this should be the 3' end, and lo and behold, that's the way that DNA polymerase is going. Notice the arrow. So we're continually adding nucleotides to this part of the strand right here, and then that makes this the 5' part. Now, one other note before we go on to talk about our next very short topic, so DNA polymerase will go this way in this replication fork. Now, this replication fork, DNA polymerase is going to have to do kind of like a leapfrog thing. It's going to have to start here and then work backwards like this, and then as DNA helicase unwinds more, it's going to start here and work backwards, and then start there and work backwards. And I'll show you that to you a little bit more as we look at the difference between prokaryotes and eukaryotes. So last topic for this video, prokaryotes and eukaryotes copy their DNA slightly differently. So prokaryotes, remember, small, simple, not very complex, they have their DNA in a circle, right? In a circular fashion. I'm not even going to try to draw a double helix, but those are our two strands of our original DNA. So prokaryotes, they're just going to have one replication bubble, and that's what we drew in the during picture in our last diagram. That circle's way too big, but that's okay. It's the same DNA. So we're going to have one replication bubble, two replication forks, right? Here's a replication fork, here's a replication fork, and so then we're going to have a piece like that and a piece like that, and then eventually we will have two circular pieces of DNA. As this continues to go around, we'll have two circular pieces of DNA with half old strand and half new strand. Alright, so that's prokaryotes. Now eukaryotes, large, complex, that's us. We're made out of eukaryotic cells. Their chromosomes aren't circular. Our chromosomes are linear, right? And we have a lot more DNA. We have about 1,000 times, probably more than that, times more DNA than prokaryotes, right? And so we're going to have a lot of replication bubbles. I'll just draw two just for simplicity's sake. But we're going to have a lot of replication bubbles, and we're going to have a lot of replication forks along our linear chromosome, right? So then we're going to have new, and then new, and then new, and then eventually it'll look something like that, right? As DNA helicase continues to go and continues to unwind, then the replication bubbles, replication forks are going to meet. And now notice, this is key, right? So here's our strand that's growing 5' to 3', right? Now what happens with this strand over here? Well, because we have to add nucleotides to 3' end, it kind of leapfrogs. So it goes here and then goes back, and then there and back, and there and back, and then same thing here. It leapfrogs ahead and then goes back, leapfrogs ahead, goes back, leapfrogs ahead, goes back. And again, that's because we can only add nucleotides to the 3' end, because the nucleotides need to bring their own energy. So it goes ahead and it leapfrogs back, and it goes ahead and leapfrogs back, goes ahead and leapfrogs back. And then eventually what you have is you have two double helixes of DNA. One double helix has an old strand, or both double helixes have an old strand and a new strand. Alright, so that's in-depth DNA replication. Our next topic is going to be how we take the information contained in DNA and turn that into proteins. How we take that information and use that information to build proteins.